In this laboratory over here, Eric has done something else for us. He's made a cell type. He's made a cell type that's cryptic. Cryptic in the sense that it has a viral genome in the chromosome. Okay, and I'll show you how he did that in a sec. But that viral genome was mutated in a bacteria by cloning so that the psi sequence was deleted. So that means this cell type has a viral genome that has all 3 viral genes perfectly, but is missing this packaging sequence. Which means in this cell type, it can make tons of the envelope protein, tons of the capsid protein, tons of the reverse transcriptase. It can make RNA, but it can't get that RNA together with those proteins. And so what Eric has done is made what's called a packaging cell line, a human packaging cell line. You can make all the proteins that you want to make a gene therapy virus, except those proteins cannot package with their RNA that was made and translated into those proteins. And that psi sequence was deleted way back here by cloning the retrovirus genome in a plasmid, deleting the psi sequence, getting the DNA, and how was the DNA then put into the helper cell? It was put in simply as naked DNA. It was just transformed like you'd add a plasmid to a bacterial cell. Very low efficiency, but if you have enough cells and enough DNA, it can be done. So you take the naked DNA, let's say the helper cell DNA, and you transform and you make this cell line. So you have it. So in this laboratory, Brian engineered in bacteria a therapeutic virus genome that had a good packaging sequence and the good ADA gene. But he also engineered this packaging DNA, which had all the genes of the virus missing the packaging sequence. And he took that DNA and he gave it to Eric, and Eric had this cell line, and Eric took it and took this naked DNA, isolated from this bacteria, added it to the cell line. It got inside by the process of transformation, low efficiency, but it got in there, and then it integrated into the genome. Elaine, are you with me? You look like you're absolutely confused to the hilt. You sure? You see the steps? Okay. So now what do you have? You have this therapeutic DNA with the ADA. You have this helper cell line, which is cranking out tons of viral proteins. And you take this DNA that Brian made, you give it to Eric, and again, you take the naked DNA, and you add it to the helper cell line. Excuse me. Just like you did to get the helper genome in the helper cell line, to make it, you take the naked DNA, this therapeutic virus DNA, add the naked DNA to the helper cell line, and it goes in. And it incorporates into a different part of the helper cell genome. So now the helper cell genome has the helper cell retrovirus genome in it, which is minus this psi sequence, and it has the therapeutic viral genome sequence in it, which doesn't have any of the viral genes but has a good psi sequence. So this genome is now transcribed into the therapeutic RNA, that is the ADA RNA, but it has the psi sequence. And because it has the psi sequence, it grabs the viral proteins, makes a capsid, it's released from the helper cell line, and there it is, and you now have a therapeutic vector. It's a virus, but the RNA of that virus is a retrovirus genome, but it doesn't have any cancer, or it doesn't have any retrovirus genes. It has the ADA gene in it. Is that clear, Tiger? What's not clear?
So you're basically just...
I'm not doing anything. There's no you here.
One is engineering the helper cell, just replacing the psi gene from the ADA into...
No. The psi is a packaging sequence.
Right.
When Brian did the engineering on this therapeutic genome, the psi sequence was left. You took out all the viral sequences, you left the psi sequence and the switches. Okay? The helper genome has the psi sequence missing because you want this helper cell line to pump out tons of proteins for the virus, but not package it with the viral genome that was contributing to making it, because if it packaged it, you destroy the helper cell line. What you're using the helper cell line for is to make viral proteins that then can surround this therapeutic genome that has the ADA gene. And the reason you want to do that is you have to infect the patient's white blood cell with a high efficiency and get this corrected gene inside those cells so that you can correct this defect. Steve, you're going to have this in discussion again, and it's also in your book, but go ahead.
Once you have the helper cell with the psi-positive packaging, it's making the packages and it has so it's making the viruses. Will that cell then start making too many viruses and then plasmolyze?
Yeah, I don't know what happens to the helper cell line, but in general, you're going to be making tons of therapeutic viruses, and so that cell line might go away, or some of the cells, but it doesn't matter because you have billions of these things, and you can keep doing it over and over and over again. All you're using these helper cell lines is to make a therapeutic virus, which is sort of like your lambda viruses, where you have the good ADA gene, you have all the things that are necessary to transcribe that ADA gene and get that genome incorporated into the white blood cells of the patient. Okay, let me move on because you're going to have this again and show you how this works. So the point here is that you get these vectors. They're now the therapeutic vector. They have the normal ADA gene in it that you can use for ex vivo gene therapy. You get the bone marrow cells from your patient that has a defective ADA gene, and you infect them with these viruses. And what happens is that the virus fuses with the membrane. The capsid gets into the cell. The RNA genome is there. The reverse transcriptase makes a double-stranded copy. Remember, there are no viral genes here. The only gene that's there in the place of the viral genes is this normal ADA gene. The double-stranded DNA finds its way to the nucleus, gets incorporated inside the nucleus as like an extra gene, and that's it. The virus can't replicate because it doesn't have any viral genes. But the normal ADA gene can be transcribed, and the protein can be made. And that protein complements the defective ADA protein. And now the white blood cell is normal because it can detoxify itself of these high levels of the purine, mora. It's really neat. Go ahead.
Doesn't it run the risk of falling in the wrong place within the nucleus, like if disrupting another
Yes,
gene?
that's a problem. That's a very good point. It runs the risk of disrupting a cell type in the patient that can cause some problems. That problem, Maura, probably set back the field of gene therapy a long time, because not with this situation, but with a very similar type of situation, there's another form of SCIDS. This SCIDS is due to this adenine deaminase gene, which is on this chromosome number 7. There's another form of SCIDS that's X-linked, and it's due to another gene that causes your white blood cells to die as well. And hold off. And so in those kinds of patients, gene therapy was used to try and treat them. And what happened was that the vector that they were using happened to land non-randomly in the genome, and they happened to land in a couple of immune response genes, and they actually caused the patients to come down with leukemia. And when that happened, all gene therapy trials of that type were stopped immediately. Okay? This was in the early 90s, because obviously you don't want to make the patients worse. However, a lot of things have changed since then. The biggest thing that's changed is the sequence of the human genome. How does that change anything? That means that you know where anything goes anywhere in the genome. You have all the knowledge in this class to be able to solve a problem. Let's say design an experiment to show where this therapeutic genome landed in the human genome. You know how to do that because you have a tag. You know what the ADA gene sequence is. You know what the human genome sequence is. You can do DNA blots or PCR. You can find what's next to the genes in the cell cultures in which it lands in. It's not like you have one cell, you have many cells. So now, when you do this, you can take the cells that you add this therapeutic gene to, and you can look at them, and you can say, where did it land? And since we know the sequence of the genome, you'd say, oh, it landed in between genes. It's not disrupting anything. And so now we have the technology to prevent that from happening because we know we can use PCR, we can use the sequence of the human genome, and we can find out where anything is that is there. We're not working blind like they were in those particular days. So now that's not going to happen, okay, because we have a lot more knowledge and a lot more sophistication. Ben, good questions.
Since the cells cannot replicate,
cells can replicate. Cells can divide.
But I mean, these cells cannot replicate the ADA producing, what's the word? Or do they like take up the gene and like do all the new white cells also produce ADA effectively?
Yes, yes, oh, okay. Absolutely. Because the genome that you've engineered is incorporated into the genome of the white blood cells.
Okay, so the therapy only needs to be done once.
That's exactly correct. Eden.
I still don't understand how the helper provides...
Hold it for after class or for discussion. You're going to have a whole discussion on this stuff, and you're going to be able to digest it, and you can look at your notes and everything else, and we'll go into really glorious detail. But I think conceptually, I think all of you, I hope, understand conceptually what's happening. Okay? So let me move on. So the point here is that it works. It works in the sense of there it is. This is one of the little girls that was engineered way back in the early 90s, but now if you want to read all about it, you can get this very recent article in the New England Journal of Medicine that documents 10 new cases done exactly the same way, and it cured 9 of them perfectly, and more very healthy, no problems, and everything else. So now it's a real powerful, established gene therapy cure. So now if you want to look, and this is published in the Journal of Gene Medicine, I'm not going to go into it, one of the latest reviews I can find, but here are all the approved, and now when I say approved, I mean approved by the Recombinant DNA Guidelines Committee, Advisory Committee, approved by institutional review boards, patient consent, and here's all the different gene therapy trials, and you can see that they're increasing, some of them decrease, but now they're increasing again for all these different diseases. It is still highly experimental. It's not 100%, and this technology is being revised over and over and over and over and over again. And here are the vectors, mostly retroviruses. That would be for what I just showed you. Here's adenoviruses, and those would be vectors for like cystic fibrosis and all kinds of other things that are being tried on a whole host of different kinds of diseases in order to be able to affect a treatment and or a curve. And I'm not going to go over that either. Now, obviously, you've picked up on this. There are some problems with gene therapy in humans. One of the major problems is the delivery system. There's not that many cells that a retrovirus can infect. So you're not going to be able to cure diabetes using a retrovirus, at least this way. You're not going to be able to cure some disease that affects your skin cells or something else. This is very good for blood cells and targets of retroviruses, but the point is that you have to be able to deliver the gene for gene therapy in a very efficient and a very effective way, and that is the major hurdle for making this a universal treatment of humans for various diseases. Gene expression levels. You can get the gene in, but maybe it's not active at a high enough level to overcome the defect. And so that's really a problem. So one has to be able to learn to engineer very high levels of making proteins in large amounts that will overcome the disease. Engineered protein stability. Proteins turn over in cells. They can be degraded. So you want to be able to have them stable once they're there. We've already talked about the mutagenesis, adverse immune reactions to the vector. There was a very tragic case, the Jesse Gelsinger case. This is another case independently which set back gene therapy. This was an individual that volunteered to undergo gene therapy trials, experimental, for a particular liver disease that he had. He went to Philadelphia to a very prominent gene therapy researcher who started treating him with engineered adenoviruses. He had a very severe immune reaction to the adenovirus and died. All gene therapy trials were halted from that point on, although now, of course, they know why that happened. There were a lot of errors, and again, human error, failure to adhere to strict NIH and IRB procedures. These are experimental procedures, but in the case of Jesse Gelslinger, there were indications that he shouldn't have been treated with this virus because of his immune system and a variety of other things. But people who do this work now are much more vigilant. This is a much more mature field. It's much more sophisticated. We have the human genome. They know what the problems are. And there's a lot of activity in this area. There's that death. A lot of activity in this area, which actually bodes a lot of promise. And of course, some of the issues, we talked about eugenics, that is improving the genome. It's another reason why germline gene therapy isn't carried out. We have to have consent, risks, regulation, all kinds of things. But one of the other things is availability to everyone. In the absence of universal health care, some people can't get gene therapy because it's too expensive, which is another reason why we have to change our health care system. Okay, would you alter the germline of your child for the traits of your choice using germline gene therapy if the procedure was 100% safe? Yes or no? No problems. You can put any gene in because you know the sequence of the human genome, get back a person, be absolutely normal, and do anything you want. Blue eyes, brown eyes, red hair, blonde hair, tall, short, whatever. Wow. Who said yes? Mike.
Why I said yes? I know that the genes that I would choose to manipulate wouldn't be any sort of
Where's the
life-altering gene. It would simply
Where's
be
Ingrid?
things.
Excuse me, Mike.
No problem.
Can you get Ingrid? Start doing her job? Go ahead.
It would be involving things that will make the child's life easier in terms of any physical defects, any things in my genome that I know have caused me problems. I wouldn't personally do the kinds of things where I would choose hair color.
But you're not concerned about maybe that gene's going to be in some Asian population 565 million years from now and it could cause some problems? I'm talking about a different population than you.
To be honest, that's not a major concern of mine, that in 56 million years, the one gene that I changed will cause that kind of problem. By that logic, I shouldn't reproduce either because the child that I have could have
Good logic, absolutely, and has them. Absolutely. Who else said yes? Max, I haven't heard from you today. You've been munching away on some gum and staring at me intently.
Yeah. So I said yes.
Louder.
I said yes because you said 100% safe, so...
So what gene would you engineer?
Just things that...
What would you do?
I would...
Wouldn't be just things. What would you engineer? Don't be vague.
I would only use genetic engineering for diseases.
Diseases?
Yes.
Okay. So if you had a child and there was some problem like cystic fibrosis or something like that, but if you had a chance to do somatic cell gene therapy and it would work just as well as germline, would you still use the germline?
Probably not, because, I don't know. Since
I can't, you're mumbling, Max.
I'm trying to think, that's why I'm mumbling.
Okay, don't mumble. Give me a straight answer, okay?
I'd probably just use somatic because because...
Okay, in that situation. What did you say, Faria?
I said no.
Okay, why?
Just because it could possibly affect future generations. Even though it is safe, whatever you decide to do now could affect the next generation.
So unknown things that could happen down the line. Who else said no? Kathy. Oh, Ingrid's back. Maybe she's our film expert. Go ahead. I can't hear you.
I understand if people want to engineer for traits that are not devastating to the child's life. Like, I mean, I would agree with that. I would want to do that for my child. But on the other hand, I am concerned with those who are looking for physical traits that aren't necessarily important to the child's health.
So you wouldn't engineer for growth hormone and make a basketball player that could get a $150 million contract and...
No.
Okay. Who else said no? Tiger said no.
I said no, and I agree in things like physical traits and things like that, because it could be passed on. And if you have like every single person engineering their child to be a basketball player, then
or whatever,
then what's going to happen? They're going to start making like 14-foot tall people, and that will ruin the game of basketball. But
no, they just make the basket higher.
Right.
Wouldn't ruin it at all.
But what Max was saying about for things like cystic fibrosis, I feel like what would be the problem in doing that in the germline? Because, if I wanted to make my child not have cystic fibrosis, instead of just doing somatic cell treatment once it was born or conceived or whatever, I would want their child to not have it either. And then if it was in the germline,
but what's the problem?
Why not just erase it from the entire
What's wrong
planet?
with that reasoning? That's a really great thing you said. Let's take that issue up. You said you'd want to remove it from the germline. That's what you said, right? And you'd want their child not to have it either, right?
Right.
Okay. That's called eugenics. That's called improving the genome of the human species. There's a guy by the name of Adolf Hitler that tried to do that in a negative way. No, I'm not suggesting that that's what you're doing, but you're going to see where I'm going in a second. Now, what I want you to do, Tiger, because I know you're super bright, we had that conversation on Thursday night. I want you to tell me the problem with what you said. You said you want your offspring, the offspring, to have it. Okay, there's the mother and the father chromosome. How many of these cystic fibrosis genes are there going to be in this child of yours that you're engineering in the genome? How many chromosomes is it going to go into?
Both? One? I don't know.
What do you think? We talked about this with the goats. I know you talked about this in discussion.
Well,
how many is it going to go into, generally?
One.
Yeah. So what's going to happen when you produce, let's say if it's a boy, sperm, or if it's a girl, eggs? One of them is going to have this gene, and one of them is not. Okay? Yeah. So that means that the children of your children have the same problem, that they may still have cystic fibrosis, because this chromosome is a cystic fibrosis chromosome. This one is the one you engineered. So that means the offspring or the embryo that gets that with another individual that has cystic fibrosis, that grandchild's going to have cystic fibrosis.
Well, it's a smaller chance, though.
Not really. It's 1 in 20. Unless they don't, if they're Northern European, it's 1 in 10. You're not going to get, you have the same problem. So you're going to have to do genetic testing. You've overcome it in one generation, but it's going to segregate. Remember, genes segregate from each other. Now, the other problem that I love that you said, absolutely loved it because you gave me such a great opening. That's why I brought up this eugenics problem. We could get rid of the cystic fibrosis gene very easily. Do you think? We just tell people who have cystic fibrosis and pass a law and say they're not allowed to have any kids. Albert loves that idea. He's just pumping away. He's Machiavellian. Do you think that would work, Tiger? Here's the individual with cystic fibrosis. We pass a law and say mandatory sterilization. Can't have a kid. We find all the people with cystic fibrosis by DNA testing and say you can't pass along your gene. So now the next generation won't have that gene, correct?
Well, it's recessive, right?
And so what is the consequence of that?
So then, you know, even if you have cystic fibrosis, if you have, you know, mate with someone
I just said you're not allowed to mate. A law was passed that you can't do it.
Okay,
What's
well,
the problem?
what if me and Brian mate and we both have recessive?
You haven't, but you haven't. You're skipping over some very important things here. Can you be more precise? Just like you were on your take-home test.
You could be
and I know you were precise on your take-home because I looked at it.
Heterozygous for the cystic fibrosis.
So if you got rid of all the people with cystic fibrosis, if you got a hold of them, you said you can't reproduce, the carriers are still going to perpetuate.
Right.
Well, we can get rid of the carriers from reproducing too, because we have genetic tests that can identify all the homozygotes and all the carriers. Now, anyone who carries one deleterious copy of the cystic fibrosis gene can't reproduce, and now you're going to get rid of it in one generation. Do you agree with that? Is that going to work?
That will work, but it will remove a lot of people from reproducing, and thus I bet a lot of those
will that get rid of the
gene?
Will that Tiger, I want you to think. I'm challenging you here.
Everyone
will that get
who
rid
has
of the gene?
A single copy of cystic fibrosis, either recessive or both copies.
None
of them are ever allowed to
reproduce. Will that get rid of the gene from ever and ever and ever and ever?
No, because it could mutate again.
Thank you. So this is BS. It's called negative eugenics. That's what people thought about doing early in the 20th century. They thought about this. I'm glad you gave me an opening. Because you with me, Faria?
Yes.
There was a very famous United States Supreme Court case read by one of the most famous jurists in American history by the name of Oliver Wendell Holmes. The case was called Buck versus Bell. The United States Supreme Court sanctioned sterilization of American citizens because Oliver Wendell Holmes said, one generation of imbeciles is enough on the basis of a state mentality test. And those sterilization laws were legal in many states of the United States and were there for many, many, many years until I think the 60s when they were overturned. Because the thought was at the beginning of the century when Mendel's laws were discovered, the thought was benevolent in a sense. Look at the human suffering by these genetic diseases. They didn't know a lot. And they thought if we could prevent these people from having kids, we could remove these genes from human populations. It was a very noble goal, actually, and that's how the movement started. But it soon got really perturbed because people started thinking about this in the context that we talked about a few weeks ago. Racial contexts, mental contexts, cultural contexts. And it had a devastating effect in United States history because the Immigration and Naturalization Law of 1928 was passed that prevented people from Asia from immigrating into the United States and from Eastern Europe because they were considered to be genetically inferior. You guys should read some of your American history. Of the early 20th century, it wasn't very, very nice. And believe it or not, and I'm not being politically correct here, as you can tell from the tone of my voice, the sterilization and genetic purity laws of the Nazis in Germany were modeled after laws that were carried out and enacted in the states of the United States of America. And when we objected to those laws in Germany, their response was, but you have laws on your books that sterilize people too for their mental capacity. Why should you criticize us? And so this whole eugenics movement with the notion of getting rid of these bad genes got off on a whole different path. And the path completely blew up, as was brought out in that little Bernowski clip that I gave you, with the National Socialist Party. And after that, the field of human genetics collapsed because no one would want to study human genes or human genetics or even touch it because of this hugely horrible stuff and really bad science that was sort of in the name of eugenics. Now, of course, all of that is gone now. And it's, obviously from things that I've been telling you in this class, it's perfectly acceptable and desirable to, let's say, augment something that's defective. But when you talk about the germline gene therapy, you're getting into now not negative eugenics, getting rid of genes. Now you're getting into positive eugenics. You're adding genes, make people taller, make them smarter, make them brighter. So that's called positive eugenics. And the problem with that is, we're going to have another vote in a second, the problem with that is that you don't know the consequences of what will happen to that gene, in human populations. Because you know that the sickle cell gene behaves in one way in a certain population, might behave in another way in another population. You know enough about how these things go from different groups, and so those are the uncertainties. That was a little tangent that I just wanted to share with you. Would you alter your somatic cell of your child for the trait of your choice? Make them red hair, blonde hair, whatever. If you knew the procedure was 100% safe, and you can do it with anything that you wanted. Now it's somatic cells and not germline. You guys done? Go. Yeah, I'm amazed. Okay. Who said yes? Wow. Let me see. Who I haven't heard from in a while? Nancy.
Hi. Hello? Oh,
Yep.
Okay. I say yes because I would choose to my child to not have any genetic disorders, and it wouldn't affect future generations. So.
But I said any trait, not a genetic disorder. Any trait.
Well,
yes, I know that you'd want to have your child not have cystic fibrosis or SCIDs, but I said any trait.
Personally, I wouldn't feel the need to give them any special trait.
You would or would not?
Wouldn't.
So who said yes for some other trait other than a disease trait? Let's change the vote. Let's just do this over again, so I'll make it a little bit more explicit. Let's just say it's how many of you would engineer your child's somatic cell for some trait that has nothing to do with disease, something that might make them smarter, look more beautiful, et cetera, et cetera. That's the key. Go. Yes and no. You voting for Rhea?
Yes.
Good. You voted yes?
No, I voted no.
Why?
Because I just don't think there's a need to do that,
Okay.
We
have Albert here. So the point is that it's okay to do it for diseases, but it's not okay to do it for traits. So in the case of the germline, you would say it's okay to do it for diseases. Maybe you change your mind now, given the context of what we've been talking about, but not okay for traits. Albert, I'll give you the floor, because you're the only one that said yes.
Well, I mean, if you could engineer your child to be smarter, that's clearly a benefit to them, or to be stronger, or faster. So why not give your child every benefit you possibly can?
How many of your parents sent you to UCLA? How many of your parents sent you to UCLA? How many of your parents are helping with your college education at UCLA? How many of you would be able to go to UCLA without your parents' help? Not very many. What if your parents said you couldn't go to college? their parents are contributing to the enhancement. It's enhancement. Do you think everyone can afford to go to UCLA? Do you think everybody can get into UCLA, but everyone can't afford it, even though it's cheaper than Harvard? I want you to think about this in the context of these kinds of issues. We'll come back to it again, Mike, because I need to go on. But the point here is that it's not absolute. If you could like take a pill, let's say, that might make you a little bit, as Albert said, get an edge to get into UCLA. I know there's a lot of people in California that would have their kids take those pills because they're just dying to get them into the UC. Stephen, one issue and then we're going to move.
There's also an element of eugenics with that too. Because
in a sense?
It would affect the sexual selection.
In a sense?
So if people prefer taller mates, then the next generation would be taller.
Yep. Yeah. The point here is that we all do eugenics when we choose our mates. That was what I was referring to with Amelia last week. We don't just go around randomly mating. We choose people. And in some respects, that is eugenics, but it's done in a way that is a little bit less frightening than the eugenic sense. So let me wrap all of this up and then move on. There are other ways to do gene therapy, and that is to deliver DNA to an individual, but deliver the DNA to an individual much like it would be sort of like a pill. The DNA then would be like a molecular drug. And what you'd want to do with this piece of DNA, it's a little bit different than we've talked about with respect to this. It is, in some respects, it could be either in vivo or ex vivo, depending on how you do it. But in some respects, what you're trying to do in this situation most of the time is shut down a gene that you don't want to be active. So in the case of the ex vivo gene therapy that I showed you with the ADA, you wanted to put a good ADA gene into the white blood cells, so you'd essentially complement, you'd complement the bad gene. But what if you have a lot of bad genes that are active, like HIV or cancer or some other viral infection that you have? You don't want those genes to be active. You want to battle them. And so in some respects, you want to shut them down. And you want to be able to, in some respects, use some forms of drugs that you can use in some way to be able to shut them down in some particular manner. So I'm going to skip that one, and I'm going to skip that one for time, and I'm going to go to that one. You've all heard of Lou Gehrig's disease. It's devastating. It's a lethal disease. It's a disease which is caused by degeneration of the muscles. It's a disease which is a defect in this superoxide dismutase gene. You all have a SOD, S-O-D protein in your cells. It's a scavenger enzyme. It's an enzyme that essentially acts as an antioxidant. There's byproducts of your normal metabolic processes that produce radicals, and those radicals can poison your cells and kill them, and the superoxide dismutase protein is part of a system that purifies your cells from these radicals as an antioxidant. So it's very important for you. And when you don't have a good superoxide dismutase gene or protein, these radicals build up in your cells, and they can build up in your muscle cells, and you essentially lose all muscular ability to do anything. And that's what the amyotrophic lateral sclerosis, ALS, does, and there's no cure for it. And it's a very devastating, tragic disease. And so it's a target for certain kinds of therapies that are very experimental, haven't proven to work, but the idea is if you could shut down this SOD gene and prevent the enzyme from doing its thing, I should say if you shut down the gene and prevent the radicals from building up, and you can be able to then, let's say, affect a cure for ALS in some particular way. And so one of the ways in which one might be able to do this in terms of that aspect is to use what I'm just going to talk about very conceptually, are these things called antisense RNAs and what is commonly known as RNA with a small I. Now, I can't get into RNA with a small I in depth because it's way beyond the scope of this class, but I'm forced to bring it up, not forced, I want to bring it up because it's probably the hottest area in human medicine in terms of gene deliveries using like gene therapy or whatever. I'll talk about antisense first. Let's say you had a gene that you wanted to shut off. That gene is transcribed. That RNA is then inside the cell. Let's say in this particular case, it's a cancer gene gone wild. It's making a protein that you don't want. That gene makes a messenger RNA. That messenger RNA is being translated and the protein is made. If you could deliver in a virus, this is a retrovirus, if you could deliver, for example, something that would make an anti-copy to that RNA, make an antisense RNA, in the cell, that antisense RNA would form a complement with the sense RNA. You'd get a double-stranded RNA in the cytoplasm, and protein synthesis would be shut down. That's the principle. And so if one could engineer this virus with a DNA such that when that DNA is transcribed, it makes a complement to this, let's say, bad messenger RNA from a cancer gene or whatever, it's going to be able to bind to it to form an RNA-RNA hybrid, and translation is going to be inhibited. The protein's not going to be made, and hopefully you'll be able to stop the course of the disease or the viral infection or the cancer or whatever. Okay? That had a very promising start in the early 2000s, but it's now been supplanted by this thing called RNAi. It's a very mystical, magical thing that would probably take me one lecture to explain to you. But the point here is that there's a system that I'll show you in a second. There's a system in all of our cells that was first discovered in plants, but then discovered in all eukaryotes, that is thought to be the genome's immune system. And what do I mean by that? You know we have these transposons that can move around, but they don't move around your genome, but they're active, they're doing things. And if there isn't some mechanism that prevents them from doing what they could do, which means jumping around, we'd all be dead. And so part of these RNAi function inside the cell, and I'm simplifying this tremendously, as Ryan knows up in Davis, and John knows, part of this system is to like shut down things in your genome that's not supposed to happen, like a transposon hopping and, let's say, mutating other genes and causing all kinds of problems with us. And so it's like a sensing system. In some respects, it's kind of analogous, and I'm stretching this, okay? It's kind of analogous to restriction enzymes I'm really stretching conceptually. It's kind of analogous to restriction enzymes in bacteria. The virus comes in, and the restriction enzyme chops up the viral genome. Well, in this particular case, this system can sense whether a transposon is active, which is not supposed to be active, and it'll shut it down. Or maybe a virus will infect your cells, and it'll shut it down. But it's sort of a defense system. Well, that defense system can be used in a very powerful way. Dr. Harada's lab uses it. We use it. It's a very powerful way for using genetic engineering for shutting down genes. And what we do is, and what's done in human gene therapy, is you make a synthetic, let's say, RNAi. And that synthetic RNAi has a target for a very specific messenger RNA. And there's a system in the cell. This is in your textbook. There's a system in your cell which can take these RNAs, put them in a complex, guide them to the target RNA, and cause the target RNA to be completely digested and degraded away. So you can use this little protection system using genetic engineering to introduce these RNAis as targets for, let's say, disease messenger RNAs or other things in order to degrade them. And this has supplanted this antisense technology. No one uses antisense technology because this is a much more powerful technology because it uses a natural system inside your cells. And whenever you can use a natural system inside your cells, it's always the best and the most powerful system, and it is a very powerful system. And so without spending a lot of time on it, there's essentially the system. You would engineer this RNA to match your target RNA by sequence complementarity. You would deliver it. Again, the delivery system is the problem here. You have to deliver it in some way. There's not a very good way to deliver it. You can use some retroviruses, but it's not very efficient, and this is all experimental, but it's hot. It goes into this complex in the cell that senses there's this foreign RNA. It has a particular target. It finds its target, and it degrades itself and essentially the target and shuts down the disease. Now, I know I'm being very conceptual, but what I'm saying is that there's ways in which you can deliver RNAs to cells by using genetic engineering that will shut down a target RNA. And if you can do that, you'd have a way of treating a cancer if you had a delivery vehicle. If you did that, you'd have a way of shutting down, let's say, HIV infection. If you did that, you might be able to shut down any viral infection. And this is the hottest area of molecular medicine. And I cannot tell you how many companies have been started that are using this technology or trying to use it to affect treatments of a whole host of human diseases. And a Nobel Prize was given just a few years ago to two young guys, Andrew Fire and Craig Milo, for discovering this particular system. Okay, let me finish this up. Here's using RNA to shut down. This is a gene that was engineered in a mouse. It's a gene that when you shine something on it, it's not the GFP, but if you shine something on it, you get a little light signal emitted. Here's the mouse that has been treated with essentially retroviruses that have these small RNAs or RNAis, and you can see that this flashing has been completely shut down. It's almost 100% effective, and that's why it's so powerful as a gene therapy. And it's been done experimentally in mice, and it's been done in mice to shut down all kinds of genes involved in cancer and other kinds of things, and it works, and it works well. But of course, it's still experimental in terms of being able to use as a gene therapy. And there are some of the countries and companies are involved. And here's essentially how it works in many respects, sort of a different system. Okay, the last thing I want to talk about are these things called ribozymes. This is a different system. This is using an RNA as an enzyme. There are some RNAs in cells that can function as enzymes. And this is why people thought that we evolved from primitive RNAs way back, billions of years ago. RNAs can have a property of being catalytic. They have the property of sort of behaving like an enzyme. Well, in some respects, you can engineer these RNA enzymes to recognize very specific messenger RNAs. You can engineer sequence by sequence because they recognize a specific sequence. And when you do this, you can take this thing called a ribozyme, you can have it match a particular RNA, and when it matches that RNA, it degrades the RNA. This is not RNAi, this is not this primitive immune system. This is a different kind of system, but again, it involves genetic engineering because you have to engineer these things and you have to be able to get them in the cells in some way by some delivery mechanism. And that's just been used just recently in a paper that was published in Nature Medicine this month or last month from a lab here at UCLA in which they used this to destroy a particular transcript of the HIV virus. They used gene therapy to introduce this ribozyme into white blood cells from patients, volunteers that have HIV, and this ribozyme then was expressed, and when a particular message was there that was necessary for the functioning of the HIV virus, it destroyed it and was able to keep these patients with a very low viral load in the absence of any drug therapy. So it's a very promising, new, novel technology. So let me just recapitulate. We have in vivo gene therapy and ex vivo gene therapy as forms of somatic cell gene therapy. In vivo, we have cystic fibrosis and treating, like glioblastomas. Ex vivo, we have the ADA example with SCIDs using retroviruses. And then we have these more experimental treatments using exogenous things like antisense RNAs or these RNAis or these ribozymes and trying to deliver them to inhibit genes rather than augment and correct gene defects. So the point here is that all of these things, in the pinnacle of genetic engineering in terms of humans, all of these things started with this experiment of Cohn and Boyer learning how to take plasmids, cut them with restriction enzymes, splice them together, and form recombinant plasmids inside bacteria. That was 36 years ago, and obviously the field has evolved tremendously with a lot of history, science, and applications. And with that, I'm going to end all of the science discussion, as I promised you, and I'm going to start on the law after I talk to you about your, well, I think I'll start on the law and then talk to you about your exam. Let me just start on the law, briefly. Just to get into where we were. Now let's go right over here. There we go. Just as an introduction. As you know, the field of genetic engineering and science has been regulated in many ways. There's all the references for this lecture, which I'll finish. And you know that the law has impacted science in many, 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 many different ways. And all of this stuff, I'm going to show you this next week. I'm not going to show you this now. All of these things really came to a head with the genetic engineering controversy in 1974 after genetic engineering was invented. And so the point is, or the question is, how can one regulate science? How can one regulate freedom of inquiry? How can one regulate experimentation? How could city councils pass laws that would say that genetic engineering can't be carried out within the confines of a particular city? How can a county in California say that you can't have any genetically modified organisms made within the context of the county? How can a state pass a law and say you can't do any therapeutic cloning or reproductive cloning? How can a nation prevent using federal funds for doing any stem cell research? How can a state or a nation pass laws and say your lab has to be inspected? The equipment that you use for genetic testing has to pass certain standards. How can a state or a country pass a law which would say, all convicted felons have to have a DNA test and has to go into a gene bank? How can a state pass a law that says every newborn child has to be tested for a battery of 80 different genetic diseases to see whether they have them or carry them? What gives the state of California or the state of Arizona or the state of Colorado or the United States of America the authority to be able to pass these rules and laws and legislate in the area of science? And what might even give them the legal prerogative to say that Albert can't even publish something that he might have found in a laboratory? Or can they do that? Or prevent Albert and I, let's say, and a bunch of you from going in my lab and doing experiments? Can that be done in the context of our system? And so what I'm going to do next Tuesday, and I hope Russ Korobkin starts on Thursday, but what I'm going to do is I'm going to go into the basis, the constitutional basis, the federal constitutional basis for how these laws can or cannot be enacted and enforced. And I think it's relatively straightforward until you get to the issue of cloning, until you get to the issue of stem cells, because it's pretty murky there. And there's not a lot of case law, And there's certainly no Supreme Court cases that one can really draw on, although Korobkin hopefully will shed some light on that. So I'm going to do that on Tuesday. And then I'm going to transition into intellectual property. And I'm going to give you a very, very nice discourse on patent law and where it stems from. And I will say before I close, there's only one mention about science in the Constitution of the United States of America. It's only one mention. It's in Article 1 of the Constitution, and it pertains to intellectual property and patents. And that's it. There's nothing else mentioning science in the Constitution, but it does mention patents because patents go back, and patents are adjudicated under federal law, under original jurisdiction of the federal courts in the Constitution. So that's what I'm going to talk about for the next couple times, and it's going to be a lot of fun, as you will see.